Cellulase enzymes are widely used in many industrial applications. One of it is in coffee processing, whereby it hydrolyzes the cellulase when the coffee beans are being dried. It is also used in detergents, cleaning, and washing agents. It is also used in paper pulp and textile industry, whereby in the textile industry it will smoothen the fabrics or the fibers of the clothes in fruit juice industry, alcohol and wine industry, whereby it is able to extract the unpleasant aroma or smells and also the tannins from the grape skins. It is also used in pharmaceutical applications industry, whereby it is able to treat the phytobezoars and cellulase bezoars, whereby the phytobezoars and cellulase bezoars are able to stay in the stomach. Cellulose are carbohydrates that are found on cell wall of plants and some microbes. Cellulase enzymes are produced by some bacteria and fungi, whereby they break down the cellulose into beta-glucose. Some humans and some mammals are unable to break down the cellulose since they don't have the cellulase enzyme, or microbes with cellulase enzyme in their digestive tracts or intestinal tracts. But some cows, rodents, and termites have the microbes which can produce the cellulase enzyme. Therefore, they are able to break down the cellulose. There are three types of cellulase enzyme. First is endocellulase, exocellulase, and cellobiase, or also known as beta-glucosidase. Endocellulase breaks down the internal bonds which interrupts the crystalline structure of cellulose, whereby the polysaccharide chain will be exposed. Then exonuclease will come in and continues to split the ends of these polysaccharide chains. The next is cellobiase or beta-glucosidase, whereby it will hydrolyze this exocellulase and thereby producing the monosaccharide structure.
Before the purification of the enzyme should be extracted and isolated from the bacteria. The first step is culture the bacteria in medium. The medium is contain the nutrients for the bacteria. Usually it contains sodium chloride. Today the cultured bacteria should be incubated for 36 hours at 37 degrees Celsius with shaking at 150 ppm. So then, after the incubator, the cultured medium go pada the centrifuge at 6 until 8,000 for 20 minutes. After that, the culture pass through the membrane to remove the cells. In order to get a purified cellulase, crude enzyme has to be treated with few purification steps, which is the first one, ammonium sulfate precipitation, and then followed by ion exchange chromatography, and lastly treated with gel filtration. Let's see how that works.
The first purification step is ammonium sulfate precipitation. Crude cellulase prepared after overnight can be precipitated by adding solid ammonium sulfate at 40 to 80% saturation. The precipitate is then left at 4 degrees Celsius for 24 hours before it undergoes centrifugation for 15 to 20 minutes. It is then redissolved in a buffer which containing sodium phosphate buffer, EDTA, and mercaptoethanol. After precipitation step, it is then followed by ions exchange chromatography on diethyl amino ethyl DEAE cellulose. In this mechanism, since the resin is positively charged, so that only anions are able to attract to the resin via weak ionic interaction. This means that enzyme with cation which is unbound to the protein will be then washed out from the column first. Weak bound protein can then dissolve as purified fraction by adding buffer with higher ionic strength. High salt solution such as hydrochloride or sodium chloride is added to iru-bound proteins. According to the journal, fractions were collected at flow rates of 0.5 ml per minute. Fractions with cellulase activity were collected and proceed to further centrifugations. The last purification step is gel filtration. Gel filtration is works on the principle of molecular size. The resin consists of beads which contain pores of a defined size range. Larger protein which cannot enter this pore will then travel around the outer parts of the beads and elute from the column first. In contrast, smaller proteins which has the ability to pass through this pore will then take a longer time to pass through the column and elute out. According to the journal, fractions were collected at flow rate of 0.25 ml per minute. Fractions with cellulase activity were collected and processed to further centrifugation. Again, precipitate were collected by centrifuge at high speed and redissolved in the buffer. Purified cellulase can be then obtained after overnight.
After the purification of the enzyme, the next process is electrophoresis. Electrophoresis is performed to determine the molecular mass of the cellulase. SDS is an ionic detergent which denature the protein. And the purpose of SDS is to create all the protein in similar charge to the mass ratio and facilitates the migration of the protein. Therefore, the separations depend on the relative molecular weight of the protein. When the voltage is applied, the protein is travels toward the positive electrode of the electrophoresis, and the polyacrylamide gel send larger molecule from the migration rather than the smaller molecule. That's why the larger molecule move slower than the smaller molecule. And the step of the electrophoresis is buffer was added to the purified enzyme and boiled at 100 degrees Celsius for 2 minutes. After that, the mixture is subjected to the electrophoresis, and the electrophoresis is performed at room temperature for 1.5 hours with 100 volt. And the protein absorbed from this process is phosphorylase B, albumin, ovalbumin, and carbonate anhydrase.
Isolate the cellulase from different bacteria to have the different characterization, such as temperature, pH, metal ion, inhibitor or retardant, and substrate specificity.
Here is the graph talk about the relative of temperature to the cellulase activity and stability. The x-axis is the temperature, while the y-axis is in black color is enzyme activity and in blue color is enzyme stability. When increasing in temperature, the activity of enzyme was increasing until its optimum temperature, which is 60 degrees Celsius, then dropped. The pH of the optimum temperature is 7. For the stability of enzyme, temperature starts from 0 and increasing. The stability is maintained in 100% until it reaches 50 degrees Celsius. Then will drop immediately. For the change of the pH, the x-axis is pH and the y-axis are enzyme activity and stability. When increasing the pH, cellulase activity and stability are very low in pH from 1 to 2 to 3. The optimum pH for cellulase activity is 6, which is in 100% activity. But for the optimum pH of stability is 7. The activity and stability of cellulase will start to drop after reach optimum of pH. Addition of metal ion will influence the activity of cellulase. When there is nothing added, activity will remain in 100%. Concentration ions start from 1 millimole to 10 millimole. Sodium ion will drop slightly, then increase slowly. For the addition of potassium ion, activity of cellulase will increase with the concentration of potassium ion. Small amount of mercury ion added will cause the activity of cellulase drop immediately. Iron 2 plus added will cause the activity drop like this curve. But the iron 3 plus added will drop immediately at 1 millimole and maintain in 50% activity from 1 millimole to 5 millimole. After 5 millimole, activity will drop again. Inhibitor and reductant are related to the characterization of cellulase. When there is only enzyme present, the activity will remain in 100%. When increasing the concentration of sodium dodeoxysulfate, the activity of cellulase will drop to near 5%. For the addition of urea, activity will increase slightly then drop slightly through the concentration. Beta-mercaptoethyl will increase the activity when increase its concentration. But addition of ethyl diamine tetraacetate will increase very little then reach to its optimum activity. Substrate specificity is one of the factors that influence the characterization of cellulase. Cellulase show highest activity against carboxymethylcellulose. Cellulose will show 100% activity when it against with CMC. Against with Abysil will give out 34% of activity. Against with cotton will give out 8% of activity. Filter paper is less than cotton, which is only give out 5% activity. And the xylens will give 14% cellulase activity.